Two neural measures differ between urban and rural song sparrows after conspecific song playback, Kendra B. Sewall and Scott Davey. Presented by Malika Ajose, Jordan Casey, Sarah Dobson, Nadia Lewis, Valeria Ortiz, and Kyra Parker.
Urbanization. In the picture on the left, the forest land has decreased between 1992 to 2010, while the urban land has significantly increased. Male songbirds living in urban habitats are more likely to be bold and aggressive in response to social and environmental changes compared to rural songbirds. Urban habitats have been recognized as impacting phenotypes of wild birds in order to respond to environmental changes. Phenotypic plasticity helps the birds cope with environmental change. Urbanization was determined by aerial images to quantify land cover in 1 kilometer squared area around each study site. Larger PC scores from analysis indicated higher abundance of buildings and paved surfaces and lower abundance of vegetation. Melospiza melodia, most commonly known as the song sparrow, was the primary bird studied. Song sparrows are most common in North America, most specifically the Northeast and Midwest. The social behavior network plays a central role in regulating patterns of social behavior. In this case, the network includes regulating male territorial behavior and aggression across a wide range of taxa. In this study, FOSS expression in the social behavior network of song sparrows from urban and rural habitats exposed to conspecific song playback. The immediate early gene, also known as IEG-FOS, allows for the identification of brain mechanisms that could be impacted by urbanization and underpin adjustments to novel environments. IEG-FOS acts as a transcription factor. Neuropeptide arginine bastosin AVT plays a central role in modulation of socio-sexual behaviors across taxa. The AVT and AVP systems are responsible for homeostatic functions including stress and osmoregulation. The AVT works within several nodes of the social behavior network to regulate sex and species-typical behaviors. AVT expression was examined within the BSTM and PVM of male song sparrows living in rural and urban habitats. AVT exposing cell bodies and bed nucleus of stria terminalis are selectively responsive to social stimuli and regulates male territorial aggression. The lateral septum plays a key role in emotional and stress response. In this study, it was expected that there would be a lower FOS-IR in the LS and PVN of urban sparrows.
Previous studies of the urbanization of songbirds found correlations between IEG expression in the BSTM and exposure to song playback, but not behavioral response to the challenge. Brains were collected longer than 50 minutes from capture, as we did in this experiment, and also previous studies used 30-minute playback instead of 10-minute playback. The objectives of this experiment were to determine how neural activation within the brain relates to urbanization, and also exactly which regions of the brain could be impacted by habitat. The methods of this experiment include the capturing of 9 urban and 7 rural male song sparrows from June 11th to June 14th, 2014, with the use of conspecific song playback. After they were captured, the brains were collected using ethical techniques. The brains were then fixed with acrolein for 4 hours and saturated in sucrose, frozen on dry ice, and stored at -80°C until sectioning and immunohistochemistry occurred. In order to catch the birds, a mist net was placed in the center of the male's territory, and one of 16 conspecific songs were played in order to simulate a social challenge to the territory holder. Songs were played at a rate of 1 song per 10 seconds at an amplitude of 80 decibels, 1 meter from the speaker. On average, it took about 8.5 minutes of playback to capture each male. Longer playback was avoided in order to avoid habituation. Blood samples were collected 3 minutes after capture. This allowed us to quantify plasma testosterone values and compare these values between rural and urban birds as well. After the blood samples were taken, the birds were held in silence and darkness for approximately 40 minutes until the brains were collected. The darkness and silence ensured no external stimuli would affect neural expression. The males were deeply anesthetized with isoflurane before being rapidly decapitated. The cryostat was used to section slices of the brain after they were fixed. Grayscale images of each region were captured using an AxioCam MR camera attached to a Zeiss Axio Imager microscope. All areas were imaged using 20x objective. The figure above shows that urban birds had a greater ABT-like staining in the BSTM than rural birds, but less FOS in the BSTM and LSC-BL than rural birds. The following areas were examined through imaging.
The results for plasma testosterone are as follows. No overall differences in testosterone levels between urban and rural birds. Testosterone levels were not related to the number of cells showing AVT-like immunoreactivity in the BSTM or PVN. The results show that rural birds, as compared to urban birds, had more FOS-IR neurons in the LSCVL and the BSTM. Habitat had no overall effect on the number of FOS-IR neurons present. However, birds that were collected later in the season had high FOS-IR, and differences in expression across brain regions were seen. It can also be noted that neither the duration of playback nor plasma testosterone levels explain the number of FOS-IR neurons in the main model. The following graphs present the main effects of habitat type on FOS-IR within 9 brain regions. These graphs account for effects of day of year, duration playback, relationship with plasma testosterone, and controls for the number of tissue sections in which we found the brain regions of interest. The results of AVT immunoreactivity are as follows. We found a significant effect of habitat on AVT-like immunoreactivity. AVT-like immunoreactivity differed across brain regions with far higher cell numbers in the PVN than the BSTM. There is an interaction between habitat and brain regions such that urban male song sparrows had more cells showing AVT-like immunoreactivity in BSTM than rural males. Although this was not seen in the PVN, testosterone levels contributed to differences in AVT-like immunoreactivity. This figure depicts the number of AVT positive cells SEM in the PVN as well as BSTM in male song sparrows collected in rural and urban habitats.
Discussion. Male songbirds in Blacksburg, Virginia have different patterns of neuropeptide and immediate early gene levels as a result of conspecific song playback, depending on whether or not they are rural or urban. Studies have found that stimulated territory intrusions increase IEG expression in the BSTM, LS, PVN, AH, VMH, CG, and VTA of territorial birds. However, aggressive response to social challenge is negatively correlated with the IEG responses in the AH, PVN, and various zones of the LS and territorial male song sparrows. In the present study, the urban male song sparrows, who are reliably more territorially aggressive, have had fewer FOS-IR neurons in the LSCVL than their rural counterparts. Although the trend of elevated FOS-IR in the BSTM of rural birds relative to urban birds, this could be because the rural birds interpreting the playback as a greater social challenge. This hypothesis can be experimented by showing different birds from multiple habitats with social changes with numerous intensities. Studies using double labeling of cells with the IEG and AVT showed that AVT neurons in the BSTM are activated by positive social stimuli, such as potential mates. Urban birds were found to have more AVT, like immunoreactive neurons, in the BSTM. AVT levels in the BSTM are associated with territorially and aggression, while AVT in the PVN is involved in maintaining homeostasis. Resolving the contribution of the AVT system to behavioral differences associated with urbanization will require double labeling cells for AVT and IEGs.
The findings of our experiment can lead to an explanation of how habitat change and neural function are related. Also, there are not very many studies that showcase the effects of urbanization on bird brains. Another question that could be raised from this experiment is how can behavioral differences between rural and urban birds and also other animals be maintained through the manipulation of neurons by their social environments? In conclusion, the experiment found that urban sparrows had reduced IEG FOS expression within the LSCVL. Also, urban sparrows exhibited more AVT-like neurons in the BSTM. And finally, urbanization does have an impact on neural processes. Critique of study. Some things that could be done differently next time or in another experiment include a longer song playback time, but not long enough to create habituation, using another technique instead of conspecific song playback, since catching birds in the wild with this method may unintentionally attract the most aggressive males of the species. Also, the examination of female birds as well, in addition to possibly testing other woodland creatures. And finally, possibly examining more drastically urban and rural areas outside of Blacksburg in order to get a better idea of the overall population.